Now, remember, on the second exam, we talked about if I then get rid of that, if I give them a drug for this, though, now guess what? There is a selective pressure to transfer this one because this one's already gone now because the drug is getting rid of it, right? Then I can switch to this one, something like that. So yeah, and one thing we're going back now is that we don't actually know. So for instance, BRCA2 is mutated in breast cancer, right? And BRCA2 is really important for what pathway? Recombination repair, DNA repair. However, every cell in our body has BRCA2, right? But it just so happens that in breast cancer, the chances of BRCA2 getting mutated is much higher than any other cell type in your body. And we don't really know why that is. So for certain genes, certain proteins, certain areas of the body are going to be more or less susceptible to mutation, and we don't know why. But that is one thing that's going to be a factor, one thing to keep in mind. Or another example is melanoma. RB never mutated in melanoma because the RB protein, basically the idea there's something to do with the gene silencing. It's very rarely RB. It's more so melanoma is what's the big one? BRAF, right? BRAF is the big one for melanoma. RB very rarely have ever mutated in melanoma. However, certain other cancer types are going to have RB mutated. What's the biggest example of that? Yeah, retinoblastoma. The retinoblastoma cancer is due to oftentimes, almost always, the RB, not then something else. I think the example in the article here was RB and P16 for retinoblastoma. I think I could be wrong. So keep that in mind that different areas of the body, different susceptibility of those genes. I'm not entirely sure why. Here's the next really big photo of random stuff. Main points, I just did 3 big things. I think those are the biggest ones for me. DNA repair, guanine methyltransferase with basically type DNA repair, and then other hallmarks of cancer. So, like we talked about before, oftentimes with DNA repair, that's going to be the first gene that's going to be mutated. Why? Because cancer is a bunch of mutations. So I have to first screw this up to then get all my other genes screwed up. So oftentimes we will lose DNA repair pathways first, like BRCA2 or MMR repair, and then after that we can then now lose other things. Hopefully, well, if you're a cancer cell, I want to hopefully lose my apoptosis genes, because then I want to make sure that even though my DNA is screwed up, I can't stop it, and I keep going through, and I get replication going on. So oftentimes these won't really be like the effector protein, right? These aren't going to be this is not the protein that's going to cause cancer. Does mutating MMR cause unrestricted cell growth? No. Mutated MMR only causes no repair. Once I have no repair, then I screw up the actual effector proteins that are going to be responsible for cell cycle, and now I have that cancerous growth. Guanine methyltransferase, this is one of the ones you had to know for that second exam. It's the one he told, it was the IRMA stuff, so it was like in between. But basically it's going to do what? It's in its name. It's going to transfer methyl groups. Oftentimes, getting a bunch of methylations is going to cause silencing of genes. Many cancer cell types are going to use this. They're going to hypermethylate a bunch of shit, or what they're going to do is they're going to screw up guanine methyltransferase. So I've now screwed up this protein, this enzyme, and therefore that protein, and now when methyl groups are there, I can't get rid of them. And therefore then what can cancer cells do? They can put a bunch of methyl groups on proteins like what? Tumor suppressors, apoptosis genes, and now I don't have those genes. Even though the genes are normal, the function is normal, I've changed that regulatory region. I've changed the area upstream of that, and now those proteins aren't going to be present. So that's what goes on with those guanine methyltransferases. Keep in mind, this is a double-edged sword. So the cancer is cancerous because it screwed up guanine methyltransferases. And then it puts methyl groups on a bunch of shit, and now those proteins aren't expressed. However, because of that, if I give that patient a methylating drug, what is that going to do? It's going to put methyl groups on all of the DNA. And then these cancer cells, they don't have this transferase because they mutated and destroyed it. Now those cells are going to die. Keep in mind, our normal cells, some of them are going to die too, just because there are a bunch of methylations going on. But our normal cells have normal good levels of guanine methyltransferase. That's why it's a double-edged sword. It is a cancer because of it screwed this up, but because of that, it's much more susceptible to methylating agents. So any sort of methylating disease drug that we give them will then lead to, hopefully, that's the cell death. Angiogenesis, once again, it's just basically the idea of blood vessels growing towards a tumor cell. Basically what happens is, like, let's say this is your inferior iliac artery, it's just going to be chilling right here. The entire thing doesn't rip off and go this way, but it kind of like sprouts out like a new little branch that wasn't there before, and then it'll go to that tumor cell. So angiogenesis is obviously something very bad. I don't know if you guys, anyone's done any surgeries, but oftentimes they can open up patients. They can assume that maybe there's some minor tumor or something like that, but they open them up. If this is so severe that there's so much blood going to the tumor, you cannot remove the tumor. You can, but you're going to kill the patient. So this is not just talking about like very small little blood vessels. We can talk about a large amount of blood can be going to this area. And keep in mind, when that blood's going there, it's not going to other areas of the body, other limbs, other vital organs as much. VEGF, once again, is that little vascular endothelial growth factor. That's the little player involved there. They can escape the immune system. Big example here are the cancers with a viral origin. So you guys probably had to know this for immuno, right? Or you're going to have to know it. Papillomavirus, herpes simplex virus 8. This one causes the Kaposi sarcoma. This one will cause cervical carcinoma. Know these. Know these, and know Gardasil. Gardasil is a vaccine for the papillomavirus. Like I said, this article is old. At this point when it was written, this is the only vaccine for cancer. I don't know if that's still true. I'm not sure, based on what you guys know now. But for the article for Dr. Blank, this is what it talks about. So it's the only one for that. We do not have vaccines for non-viral origin cancer. So what this means is, this is a virus that comes in. That virus then going to screw up the DNA, cause unrestricted cell growth. For those two examples. Know those examples, know what disease they cause, and then know the example of Gardasil is a drug that's a vaccine for the papillomavirus. Earlier in, I think, this exam, we talked about the fact that we can mutate and change MHC or get rid of MHC on cancer cells. What does that mean? Now it's even harder for the other, our immune system to try to identify those cells at all to attack and destroy, things like that. This literally is the same thing. The RAS pathway with your BRAF, where you have your RAF, MEK, and ERK. This is that same thing for the 8th time in this lecture. So make sure you at least understand that fairly well. More about it. In particular, now we're focusing on the receptor. The ligand for it is going to be a growth factor. There's like a bunch of different kinds of growth factors. No, it's a general growth factor. In particular, when it binds, it's going to cause a receptor dimerization. So that's where you had the thing like this. The signal comes in, and they come together. And the inside, they're going to fiddle around, and that's going to cause these downstream effects that are going to be occurring. Receptor dimerization leads to cross-phosphorylation. So these tails on the inside are going to phosphorylate each other. Doing that then leads to those effects with the RAS and then the BRAF and all that stuff downstream. Excuse me. Yeah, in particular, this is the general receptor, but even different cell types, there can be slightly different variations in terms of where the proteins are located, if they're physically part of the receptor on the intracellular side, or if they're proteins just around that area. It's kind of this sort of, depends on the type, the area we're looking for in particular. And this is the same thing before, downstream effect of this entire RAS pathway, RB CDK4, cyclin-dependent kinase 4, that leads into other pathways, therefore leading into the other changes that we're going to see. BRAF is the one form of RAF. In particular, downstream effect at the genetic level is going to be AP1. So AP1 is a transcription factor, important for cell proliferation. So basically, the end result of going through that whole pathway, I increase AP1. AP1 is a transactivator. So what that means is now in my DNA, this AP1 can bind to this response element, and these genes right here are ones that are going to be for cell growth. So that should make sense. I have more TA, I get more of this gene. These genes are for more cell cycle. Therefore, we get the unrestricted cell growth if this pathway is always active. Don't forget the BRAF mutation, V600E. I think it's vimurafenib. Yeah, vimurafenib, V600E. That's the mutation. This is one of the first examples, not anymore, but one of the first ones when it came out, of a drug that targets a protein with a specific mutation. Other drugs we've talked about, they target all of that protein, which means that the bad protein in the cancer cells, the good protein in our cells, or the good protein in our normal cells that are healthy. This vimurafenib only is going to target BRAF with a glutamate where the valine should be. So it's much more specific, much better in terms of we have much fewer side effects than a lot of these other cancer drugs that we're talking about. T-cell leukemia, an example of when RAS pathway is mutated. This actually was for the second exam. It was the example of that pathway, when it's mutated. If the RAS pathway is mutated where it's always active, you get T-cell leukemia. The target gene is interleukin-2. So the target gene at the end of that pathway in T cells is going to be interleukin-2. That's the autocrine factor. That's where the T cell releases interleukin-2. It can come back to the same cell, or go to other T cells, and those T cells constantly replicate. And that would then lead to that lymphoma, T cell leukemia in that example. Normally, interleukin-2 is only going to be made when there are foreign pathogens, because then it's going to bind to the T cell, and we get the interleukin-2 getting shut up. But in this case, I mutated the pathway where that even though there's no signal, that pathway is active, and I make a lot of interleukin-2. Therefore, I'm going to constantly be proliferating and making a lot of T cells. JAK-STAT pathway, one of the ones from the second exam. This one's the easiest one. You've got basically the receptor. It's going to have the signal bind to it. You basically phosphorylate STAT. STAT's going to dimerize, and basically the STAT then acts as that transcription factor. This is the example of where those phosphates are on the inside of the proteins. Why is that important? Because phosphate's negatively charged, DNA is negatively charged. So with this dimerization, these phosphates are on the inside, and it doesn't matter that they're negative. This thing can still go in and then bind to the DNA. Translocations. This is another good example because Blank loves diseases. So be aware of these translocations. So what this just means is that basically it is where parts of one chromosome end up in another chromosome where they shouldn't. And thinking about this, what do you think, what pathway or process that we talked about today will prevent translocations? It's a type of DNA repair. What's the big one? Recombination repair. Recombination repair is going to be one that's going to prevent when I have these crazy, because what happens in recombination repair, what kind of repair is it doing on DNA? Single-stranded breaks or double-stranded breaks? Double-stranded breaks. So if I don't repair those, I got shit flying around everywhere, and it can migrate and move somewhere else. So RR, recombination repair, is going to prevent this spaghetti stuff from inserting itself somewhere else. So if I lose recombination repair, I have a much higher chance of a translocation and protein fusion genes from occurring.
Did you say BRCA2 was?
BRCA2 and RAD51 are very important for recombination repair. You should know both of those for recombination repair. So we've got translocations, and then we have fusion genes. They're slightly different. Well, a fusion gene is, I believe it's a more specific type of a translocation. In general, translocation means a bunch of things from one chromosome gets inserted into another chromosome. Within there, though, the insertion where it occurs is really important. In a fusion gene, it literally is where this gene and this gene are together. So when I make the protein, it's this crazy abnormal protein that we haven't seen before. So the fusion is two genes together, and they get smushed in. There's no intron in between them. There's no random space between them. Or there could be an intron, but it gets spliced out. But literally, these two genes have now become one gene. They are fused together, and the protein is a new protein. Protein literally is half the normal gene, half that gene, and half this gene together.